Assalamu alaikum wa rahmatullahi wa barakatuhu. The title of this lecture is Amino Acid Derived Biomolecules. From this lecture, you should be able to understand and describe the use of amino acids in the biosynthesis of important molecules such as creatine, glutathione, porphyrin, serotonin, thyroid hormones, epinephrine, purine and pyrimidine, carnitine, S-adenosyl methionine. This lecture is divided into two parts. In part 1, the upper three, creatine, glutathione, and porphyrin, will be discussed. The content of this part will be biosynthesis of creatine, biosynthesis of glutathione, and biosynthesis of porphyrin. Amino acid not only provide the building block for proteins, as you have already learned in GM1. In addition to that, they are precursor for various compounds which has important functions in our body, such as high energy compounds, enzymes, hormones, porphyrins, neurotransmitters, purines, and pyrimidines. This slide shows various biomolecules derived from amino acids. Here in this table, you can see various compounds produced from precursor amino acids and their functions. Arginine is the precursor for nitric oxide, and the function of nitric oxide is to cause vasodilation. Arginine and glycine together produces creatine phosphate, which is an energy store in muscle. Glutamate produces gamma-aminobutyric acid or GABA, which is a neurotransmitter. Glutamate, cysteine, and glycine together produces glutathione, which is a redox agent. Glycine and succinyl-CoA is the precursor of porphobilinogen, which is the precursor for pyrrole rings. Histidine is the precursor of histamine. The function of histamine is to cause vasodilation. Lysine is the precursor for carnitine. The function of carnitine is fatty acid transport. Methionine is the precursor of S-adenosyl methionine. The function of S-adenosyl methionine that it acts as a cofactor in various metabolic reactions in our body, and it donates methyl group to produce new compounds. Here you can see that tyrosine produces so many compounds. So the number one is dopamine, or tyrosine is the precursor of dopamine, and the function of dopamine is it is a neurotransmitter. It produces epinephrine, which is a hormone, norepinephrine, which is a hormone as well as neurotransmitter. It produces melanin, which gives the pigment color of our skin. That's why our color, due to this melanin, our color varies from person to person. Tyrosine also produces, or tyrosine is the precursor of thyroxine, which is hormone. Tryptophan produces melatonin and serotonin. Melatonin is a hormone and serotonin is a neurotransmitter as well as a vasoconstrictor. The first one we will discuss is creatine. It is present in the muscle as creatine phosphate or phosphocreatine. It is a high energy compound, can reversely donate phosphate to ADP and forms ATP. Provides a small but rapidly mobilized reserve high energy phosphates to maintain intracellular level of ATP during first few minutes of intense muscular contraction. Biosynthesis of creatine phosphate. Creatine is produced from arginine and glycine. In this reaction, SAM or S-adenosyl methionine acts as a cofactor. Did you remember I have shown you in the previous table that SAM acts as a cofactor in various metabolic reactions in our body. So this is one of the examples. And the conversion of creatine phosphate from creatine is catalyzed by the enzyme creatine kinase. This creatine kinase is present in the muscle cells, so it helps in the formation of creatine phosphate in the muscles. But when there is muscle damage, this creatine kinase is released from the muscle into the blood, and there is high level of creatine kinase in the plasma. This creatine kinase level in the plasma is also increased in myocardial infarction, which is a damage or infarction of the cardiac muscle or heart muscle. Creatine and creatine phosphate spontaneously degrades to creatinine at a slow and constant rate. Then, through the circulation, it goes to the kidney and excreted through the urine. Creatine excretion is proportional to the total creatinine phosphate, which is proportional to the muscle mass, because creatine phosphate is present in the muscle, right? So if muscle mass decreases due to malnutrition, due to paralysis, or any diseases which cause muscular dystrophy or atrophy, then there will be definitely less creatine phosphate content of the muscle, and then the urinary creatinine excretion will also be low. And as this creatinine is excreted through kidney, so if anybody has impaired kidney function or kidney failure, then this creatine excretion is impaired. Glutathione synthesis will be discussed in this slide. Glutathione is tripeptide gamma glutamyl cystinyl glycine. It is synthesized from L-glutamate and cysteine, producing gamma glutamyl cysteine by the enzyme glutamyl cysteine synthesis. And the rate limiting step is regulated by cysteine availability. After that, glycine is added to the C-terminal of gamma glutamyl cysteine by glutathione synthase. Most important function of glutathione is it is a reductant which is crucial to maintain the stability of erythrocyte membranes. In our body, radicals and hydrogen peroxides are continuously formed as byproduct of metabolism. So this hydrogen peroxide attacks the double bond in phospholipid and makes the cell membrane rigid. This glutathione, reduced glutathione, is important in reducing hydrogen peroxide to water so that no more hydrogen peroxide is available to damage the cell membrane. Another function of glutathione is to transport amino acid across the cell membrane and release into the cell. So in this image, you can see when the amino acid comes near the cell membrane, then glutathione comes here and binds with the amino acid, forming gamma glutamyl amino acid. So this one then releases amino acid within the cell. Another important function of glutathione is to conjugate with drugs to make them more water soluble for excretion. Here you can see glutathione is binding with thiopurine, which is an anti-cancer or anti-metabolite drug. And with binding with thiopurine, it is forming thiopurine glutathione, which is water soluble and helps the excretion of thiopurine through the kidney and reduces the toxicity of the medicine in the body. Porphyrins are a group of heterocyclic organic compounds. They are composed of four modified chiral subunits interconnected at their alpha carbon atoms via methylene bridges. Some of the iron-containing porphyrins are called heme. Heme-containing protein or hemoprotein are found extensively in nature. Hemoglobin and myoglobin are the examples of two oxygen-binding proteins that contain iron porphyrin. Various cytochromes are also hemoproteins. In this slide, you can see the synthesis of porphobilinogen, which is the precursor of pyrrole ring. Did you remember that porphyrins are composed of pyrrole rings? Okay, so in the synthesis of porphobilinogen, we need the amino acid glycine. Here you can see the glycine. It combines with succinyl-CoA, then it forms ultimately porphobilinogen by several steps of reactions by converting to alpha amino beta keto adipate, then delta amino levulinate, then two molecule of delta amino levulinate forms porphobilinogen, or the first precursor of pyrrole ring. Another important point here, I want to emphasize that this reaction needs pyridoxal phosphate, which is a vitamin B6, as cofactor. In this slide, you can see the synthesis of hemoproteins from succinyl-CoA and glycine. So after forming porphobilinogen, as you have seen in the previous slide, then through several steps, it forms heme, and then ultimately heme proteins. This will be discussed in details in HNL module in year 2.